All right, so in the last lecture, we talked about the RAS, activation of RAS by growth factors and growth factor receptors. So this is just a kind of a summary of what we talked about in the last lecture. This slide is not on your handout because I didn't put it out, but it's a very simple slide. So the example we've been using is EGF, but it works for PDGF or NGF or a whole list of growth factors that were on your handout. So the binding of growth factor activate receptor tyrosine kinase, and that allows dimerization and autophosphorylation of the receptor tyrosine kinase on itself in an intermolecular manner. And that create a whole slot of phosphotyrosine residues. And those phosphotyrosine residues can serve as docking site for the binding of proteins that have SH2 domains. And these are the proteins. There's several examples of proteins. There's GRAP2, phospholipid C gamma, PI3 kinase, which we're going to talk a little bit more in the later lectures. And we mostly focus on this pathway. We talk about the EGF receptors having phosphotyrosine allows the binding of adapter molecule called GRAP2, which essentially has one SH2 domain that binds to the phosphotyrosine here, and two SH3 domains that binds to this guanine nucleotide exchange factor, GEF, called SOS. So this interaction allows the SOS to be recruited to the plasma membrane where the RAS protein sits, and RAS is a small GTPase. Upon binding of SOS with RAS, that leads to the binding of GTP to RAS and leads to its activation. So today we're going to talk about how does RAS act, what's the consequence of RAS activation. And okay, so before we go to that, let's discuss a little bit more about the cancer mutations. So there are two key components in this pathway. As you can see, one is the tyrosine kinase receptor itself. The other one is the RAS protein. These are the two molecules that has catalytic activities. These are two, this is the adapter molecule, and this is a GEF protein. But these two are the key catalytic component of the pathway so far, and therefore they are the key target of human cancer mutations. So this receptor tyrosine kinase pathway is very important for regulation of cell growth, and therefore in human cancer, cancer is essentially the problem of cell proliferation out of control. And therefore, when you have cancer, many times this pathway is turned on constitutively. And the way the cancer cells turn on these pathways is by introducing mutations. So the major target of mutations are in the RTK itself or in the RAS. About 30% of human cancer nowadays that we know have mutations in RAS, and that resulted in its constitutive activation. And we're going to talk a little bit more about that. So the consequence of a lot of these mutations is to lock the pathway in the on state, so that even if you don't have a growth factors, all these pathways are kept in the on state. So the cell seems to receive the proliferation signal all the time, so they proliferate nonstop. Now, there are also, so one of the things I'm going to discuss a little bit more is the a receptor mutation in the protein called HER2. But before that, let me just finish this part. So there's a small set of lung cancers have activating mutations in the EGF receptor itself. Those point mutations result in constitutive activation of EGF receptor, and that leads the whole pathway to be activated. And so there are specific drugs, bless you, that inhibit the EGF receptor kinase activity. There are two of those drugs that you probably have heard about. This one is called Iressa, and the other is Tarceva. Iressa has been pulled out by the drug administration because most of the people here in the Western world do not respond very well to Iressa. But in Asian populations, in the Japanese study, about 40% of lung cancer patients there actually respond very well to Iressa. So Tarceva is a newer replacement of these, and it also target EGF receptors. So it's very useful. Now, another variation of the EGF receptor is the HER2. Now, HER stands for human EGF receptor family. Okay, so HER2 is essentially the human homologue of EGF receptors from mouse. So we called it HER. Now, this family are also receptor tyrosine kinases, and they bind to the growth factors, epidermal growth factors, in the growth factor in humans. So there's several versions of that growth factors. There's EGF, there's TGF alpha, and there's heparin-bound EGF, and there's several other molecules. So you don't need to know the details of these. All of these growth factors can bind to their own corresponding HER receptors. And in human, there are 4 HER proteins. There's HER1, HER2, HER3, and HER4. Now, HER1 and HER3 and HER4 can all bind to ligand as shown here. Okay, so these are the ligand that bind to HER1. These are the ligand that bind to HER3 and 4. The only exception is the HER2. And this is the protein that's really interesting. So these receptor tyrosine kinases, it's just like the EGF receptor we talked about. They bind to ligand, they can dimerize, they can get activated, and then they can initiate downstream signaling. But HER2 does not bind to ligand by itself. So it relies on the other 3 HER receptors. What it can do is to heterodimerize with any of the other HER proteins. Okay? So, for example, when you have this ligand binding to HER1, HER1 can homodimerize. Homo means 2 identical receptors get together, just like the case we talked about. But HER2 can also dimerize with HER1. And because the ligand binds to HER1, and that leads to dimerization with HER2. And this also allows receptor activation and initiation downstream signals. Same thing, HER2 can also heterodimerize with HER3 and HER4. In every case, this dimerization is ligand dependent and can also lead to activation of tyrosine kinase activity of both HER2 and HER3 and can send signals downstream, okay, and leads to cell proliferation. So this is the interesting property of HER2. Now, if it doesn't bind to ligand, why is it there? We don't quite understand the normal function of HER2. But what we know is that in normal breast epithelial cells, the level of HER2 is actually quite low. But in breast cancer cells, in about 25% of breast cancer cells, HER2 level is very high. And the reason the level is high is because the gene locus that encode the HER2 protein is being amplified, meaning that instead of having 1 copy of this gene, the breast cancer cells have many copies of this gene. So as a result, when you have that many copies of genes, each of them can be transcribed to produce mRNA to produce proteins. You end up having a lot of proteins. So in 25% of the breast cancer patients, they have overexpression of the HER2 protein. And the consequence of that is when you look at the survival curve, so the x-axis is the month of detecting breast cancer, and the y-axis is disease-free survival probability. So at the beginning, of course, all the patients start out by living, so you get 100% survival, essentially. Now, if you look at the patient without HER2 amplification, that is, the HER2 level is still very low, the patient eventually they can live up to 6 years and still have about 50% of patient survival. But in the population that has HER2 amplification, you see that the survival dropped slightly over a year to less than 40%, but probably 30%. So the prognosis is really bad. Those patients that has HER2 mutations live much shorter life and suffer much more. So the interesting part is people don't quite understand what caused the HER2 mutations. As I said, high HER2 indicate poor prognosis and shortened survival. So there's a drug called Herceptin, and many of you have heard about this. It's a monoclonal antibody against the tyrosine kinase activity of HER2. And so when patients take that, they can inhibit the activity, and then they can reduce the recurrence by about 50% and prolong survival. And this HER2 mutation, as I said, we don't understand why it happens in some population, why it doesn't happen. For the moment, we still need to do more research on that. One of the interesting features that I read is that in Asian population, especially Asian women who live in Asian countries, the breast cancer rate is actually much lower than the breast cancer rate in Western countries. However, for those patients that get breast cancer over there, a much higher percentage of them have HER2 amplifications. So the prognosis is actually pretty bad. And the age that they have breast cancer is actually earlier. So yes, the overall population has a lower rate of breast cancer, but if they get it, it's actually worse. Nobody understands the reason. So why does HER2 make the breast cancer so bad? And the reason is that the HER2 can dimerize with everybody. Okay, so when you have low level of HER2, these receptors mostly homodimerize, then they depend on a certain concentration of growth factors in order to get activated. But when you have a very high level of HER2, it's there ready to bind to everybody. So all you need is very low level growth factors. HER2 is able to sensitize all the other receptors to lower concentration of growth factors. So when you have a lot of HER2, all the other receptors become activated much easily at a much lower growth factor concentration. And therefore, the cells now start to proliferate at a concentration of growth factor where in the past it wouldn't really proliferate. But now they will all proliferate just because HER2 is there to sensitize everybody. So this is the reason why HER2 is actually bad. And that's why targeting HER2 is a very good way to try to treat breast cancer. So that's one example of we can target EGF receptors or HER2, these RTKs to be used in the clinical treatment of cancer. The other major target of cancer mutation is the RAS protein. And usually the mutation results in constitutive activation of the RAS GTPase activity, of the RAS GTP binding form. And the way it's doing that is a lot of mutations target this amino acid residue 12 in RAS. This residue 12 is normally glycine, but then the mutations can replace the glycine with a valine residue. And this replacement locks the RAS in the GTP bound form that cannot hydrolyze GTP, and therefore it's constitutively active. The RAS-D mutation that we talked about in the last lecture, it's one of these. It's not exactly the same mutation, but it's a similar mutation that target a key residue and lock in the GTP-bound form. And so these mutant cells that has the RAS mutation will proliferate as if there's growth factors, where in fact there's no growth factors, but the cells keep proliferating. So these are the list of human cancers that have the RAS mutation to show you how important this RTK RAS pathway is and how widespread. It's pretty much exists in every cell type, and that's why we choose to discuss this pathway. Okay, so this is, again, the same summary of the pathway, and we will see that activation of this receptor, tyrosine kinase, not only result in activation of RAS, but also result in change in cell metabolism, calcium signaling, and we'll see later on that it will also result in cell survival and cell growth. So what is the consequence of RAS activation? So in the case of a G protein coupled receptors, we talk about that the activation of the effector protein create a second messenger, which then activate a protein kinase, right? That seems to be the common theme. And here's the same thing. So RAS GTPase activate a protein. It's not just one protein kinase, but actually a series of 3 protein kinases. We call it a kinase cascade called the MAP kinase pathway. It involves essentially 3 kinases. The most downstream one is called the MAP kinase. MAP kinase stands for mitogen activated protein kinase. So that's where the name MAP comes from. MAPK has another name. It's also known as ERK. Sometimes you hear about ERK. Stands for extracellular signal activated kinase. It's the same molecule. Okay, MAP, K, and ERK is the same molecule. So the most downstream kinase is the MAP kinase, and you need another kinase to activate this kinase. People call it MAP kinase, or we give another name, MEK, M-E-K. And then that MEK need activation by another kinase. It's sometimes people call the MAP KKK, MAP kinase. And that name sometimes is replaced by a name called RAF. Okay, so it's essentially a kinase cascade. You start with one, and then that activate the whole thing. And what RAS does is to activate the first one, which is RAF. Okay, so this is how it works. So this is the same thing, cell membrane. That's the step we talked before. Ras get activated and bound to GTP. Now, in the cytosol, the first kinase in the cascade is a RAF kinase. This kinase normally exists in the cytosol and is kept in the inactive state. So the protein, it's one polypeptide chain, okay? But it has two major domains. One part is the N-terminal part of the domain. It has a regulatory function, and the C-terminal part has a kinase activity. And this is the serine-threonine kinase, okay, not a tyrosine kinase. So normally, the N-terminal part of this molecule fold back, bind to its strong C-terminal part, and that keeps this kinase inactive. And you don't need to memorize or worry too much about the 14-3-3. It is another cytosolic molecule that after this inhibitory complex formed, the N-terminal subunit bind to the C-terminal subunit, 14-3-3 bind to both of them and stabilize this complex. So as long as you understand, that's fine. You don't need to memorize this. You just need to know that the RAF, the N-terminal part of the molecule, bind to the C-terminus and inhibit its function. So when RAS get activated, RAS-GTP can bound to the N-terminal regulatory domain right here. And that binding, number one, recruit RAF to the plasma membrane region. And number two, it results in a conformational change so that the C-terminal domain is now free to act as a kinase. So it's activated kinase. Okay? So the way we know that this is the case is when people make a mutation by deleting the N-terminal part of the RAF, the result C-terminal half of the RAF is constitutively active. So that even if you don't add growth factors, RAF is constitutively active. All the signal cascade downstream of RAF is activated, even if you don't add signals, even if you don't have RAS. So we know that the N-terminal part of the molecules is important for inhibiting its activity. So this is how the first kinase is activated. Now, once RAS-GTP bound to the regulatory subunit, the C-terminal half of RAF is activated, the GTP get hydrolyzed, RAS go back to the GDP state, and RAF can then move on to do the downstream step. So as I said, the RAF is a serine-threonine kinase. That's the first kinase in the cascade. The second guy in the cascade is a protein called MEK. MEK is a MAP kinase, okay? Or MEK stands for MAPK and ERK kinase. I mean, these two are kind of same molecules. They call it MEK. MEK molecule, it's pretty simple. It's normally inactive. And when RAF phosphorylate MEK, that make the conformational change, turns it into an active kinase. So what's unique about MEK, which is different from all the other kinases that we've seen so far, MEK is a dual-specific kinase, okay, meaning it can target both tyrosine and threonine residues. In the past, we talk about PKA, PKC, KEM kinase. These are all pure serine-threonine kinases. They phosphorylate serine or threonine, but not tyrosine. And then we have RTKs, the receptor tyrosine kinase, that only phosphorylate tyrosine on tyrosine residues. And this was the first kinase that people isolated, knowing as a dual-specific kinase. There are a couple more, but not that many. So this is one that we're going to talk about, that we actually learned in all the courses. So it's a dual-specific kinase. It phosphorylates tyrosine and threonine. And we know already the protein target of MEK is MAP kinase. So MAP kinase activation needs two phosphorylation steps. The first phosphorylation step is phosphorylation on tyrosine residues. Okay, so this is the inactive MAPK. Without any signals, the MAP kinase is also a cytosolic protein. The catalytic center of the MAP kinase is usually bound by a little lip region, they call it, a little stretch of amino acid that bind to it and then essentially cover it up so that this region cannot bind to ATP and cannot act as a kinase. That's how it's kept in the inactive state. What MEK does is to induce phosphorylation on tyrosine first. And that phosphorylation on tyrosine then exposes the threonine side. that allows the MAP to phosphorylate the next threonine. So 2 phosphorylation events have to happen, 1 on tyrosine, 1 on threonine. When that happens, it allows 3 major type of changes. First of all, this lip region that usually covers the ATP binding site slip away, okay? And that allows the ATP to bind to the catalytic site. A protein kinase essentially take the phosphate from ATP, add it to a residue, so it needs to bind to ATP. All protein kinases bind to ATP. Without able to bind ATP, the kinase is dead, okay? So the first step is to remove that inhibition, allow ATP to bind to the catalytic site. The second is that now it binds to it has create a phosphotyrosine, and we know phosphotyrosine can bind to SH2 containing proteins. So now what it does is to allow the substrate of MAP kinase to be recruited to the close proximity of the kinase through this SH2 phosphotyrosine interaction. Okay? And then third is to allow dimerization of MAP kinase. Now that it changed enough conformation, it dimerized, and that's an active kinase as well. So this is how the cascade get activated. So you start with RAS, it activates RAF, that goes to MEK and goes to MAPK. So that's we call the MAP kinase cascade because it involves 3 kinases. And when we talk about Ras-MAP kinase pathway, that's what we're talking about, how the RAS activate RAF, MEK, and MAP kinase. And this cascade exists, as you see, in many forms in pretty much every cell type in our body. So what is the consequence of MAP kinase activation? So for now, we see there's so many steps going from the top to the center, and we haven't even touched the cell proliferation yet. And everything is still in the cytosol. So MAP kinase is a place where they start to go into the signals, start to go into the nucleus. So the dimerized form of MAP kinase can translocate into the nucleus, where now finally it's touching the transcription machinery. Okay, so growth factor stimulation, we said it induces cell proliferation, and cell proliferation means 1 cell becomes 2. And that's a big action. It needs the cooperation or coordination of many proteins. So, and then a lot of times when these things happen, you need to synthesize new proteins. You need to synthesize precursors for DNA synthesis and DNA replication. So all of these genes need to be transcribed and turned on. And so in the growth factor stimulation induces expression of over 100 genes before cells even start to replicate its DNA. So a lot of things have to happen before the cells are ready to split. And these genes are called early response genes because they react very fast. Some of them react as early as 20 minutes after the addition of growth factor. And at this point, the DNA hasn't replicated yet. So these are early response genes, and there are many early response genes. These early response genes can do many things. First of all, many of them are required for DNA replication. Many of them are required for the increased volume of the cells. Some of them are required for the expression of what's called the late response genes, that you need to generate these proteins first, and these proteins then act to activate another set of gene expression we call the late response genes because it takes a longer time, takes hours or even days for these genes to be turned on. And so these early response genes act on these late ones to increase the expression of them. So it's a tiered action. So I'll give you one example of these early response genes, and it's called FOS. FOS is also a very important gene in cancer because it's all discovered from cancer as an oncogene. That's why I'm printing it out. But there are over 100 genes like this. So I'll use FOS as an example to show you how MAP kinase activation can turn down FOS expression. So as I said, it's early response genes, meaning that the genes need to be transcribed. And how does gene get transcribed? What's responsible for gene transcription? Transcription factors. Okay, so MAP kinase is not a transcription factor. So MAP kinase need to activate transcription factors. So we start with CFOS promoter. Okay, so CFOS, in order for its transcription to happen, the promoter need to be bound by enhancer and transcription factors. So this is what the FOS gene promoter looks like. This is the coding region of the FOS gene, and this is the transcription initiation site. upstream in the promoter region, C-Fos gene has this DNA sequence. We call it serum response element, or SRE. The reason it's called serum response element is because every time you add serum, it gets activated. The transcription starts. What's in the serum? Growth factors. So this is essentially growth factor response element. That's a short stretch of DNA. Now, this DNA element usually, when the transcription happens, it needs to be bound by 3 proteins. 3 proteins, there are actually 2 types. One is a dimer of SRF, serum response factor. Serum response factor binds to serum response element. So it's not terribly creative. So it's a dimer of serum response factor and another protein called TCF, ternary, what's C? Ternary complex factor. So these 3 proteins form a trimer. Once they form a trimer, they combine to SRE and then recruit other transcription activators to start transcription. Now, normally these 3 proteins are not bound to the promoters, obviously. They need, the only way to make them bound to the promoter is that they need to be phosphorylated. Okay. So what phosphorylates these 2 proteins? Now, MAP kinase form a dimer, goes into the nucleus. In the nucleus, MAP kinase can directly phosphorylate TCF. So MAP kinase is the direct kinase for TCF. Now, MAP kinase cannot phosphorylate SRF, the serum response factor. It needs to go through another kinase. So MAP kinase phosphorylate in the nucleus, phosphorylate this kinase called P90RSK. P90 means a protein of 90 kilodalton, and the name is RSK. I can't remember the RSK, what it stands for. But essentially, it's just another kinase. Okay? So MAP kinase phosphorylate RSK, activated, and this kinase then in turn phosphorylate serum response factor. So then now TCF, SRF, upon phosphorylation can form a trimer, bind to the DNA sequence response element, and activate transcription of C-Fos gene. And of course, similar idea, similar concept happens to allow activation of many other transcription factors that need phosphorylation to be activated. And those transcription factors can then bind to individual promoter element, their favorite promoter element, and activate all of these hundreds of early response genes. So here we have the whole pathway. It's a pretty long pathway. You start with growth factors, bind to the receptors, going through the adapter molecule to recruit the GEF in order to activate RAS. And RAS activate the MAP kinase cascade that I highlight in the shade, RAF, MEK, and MAP kinase. And MAP kinase is eventually phosphorylate and activate transcription factors to allow early response gene transcription and allow the cells to be prepared to go into cell cycle. So when we study these signal transduction pathway, how do we know the relative position of each other? How do we know RAF is behind RAS? So the way that we can do that in the lab is to play tricks using mutations. For example, you see that this is a linear pathway, right? Of course, in reality, nothing is linear, but in MCB 130, things are linear. Okay, so this is a linear pathway, and as you can see, you can think logically. If I block RAS, would the growth factors cause cell proliferation? No. So you can block any step in the middle, and then you can block the whole pathway. Any events that happen before the step you block should proceed as normal, right? And any step after the step you're disturbed should not proceed normally. Now, the other way to do it is to introduce constitutive active mutations, like constitutive active RAS. If we put it in, you don't need anything ahead, right? The pathway can still go through. So these are the tricks that we do in the lab. For example, in order to figure out the relationship between RAS and RAF, which is upstream, which is downstream, people use generate mutations to inactivate the kinase activity of RAF. So now they add growth factors. Of course, the pathway has no response. Now they add a constitutive active RAS. Now, if RAF is ahead of RAS, is upstream of RAS, active RAS should induce cell proliferation, right? If it's downstream, like what I drew now, Ras will not do anything. So that's the type of experiment people did. They add constitutively active Ras, no response. So they know RAF must come after Ras. All right. So the other thing they did is to introduce an active RAF, and now the cell can proliferate. Even without Ras, even with a constitutively negative, a negative inactive Ras mutant, or a kinase dead receptor tyrosine kinase, none of these matters because a downstream step, RAF, is activated. So this is how we know exactly where the positions are, and the same way we figure out how the MAP kinase, and all of these sequence of events, how things happen. So this is a pathway. Now, as I said, life is never simple. We talked about G protein signaling before, and how G protein coupled receptors activate G protein to go through cyclic AMP and all these second messengers. Now I'm going to tell you that the Ras-MAP kinase cascade, especially the MAP kinase cascade, can also be activated by G protein coupled receptors. And one example is that studied the best is the yeast mating type pathway. So in yeast, we know there are two mating types. There's alpha and there's A. These are all haploid cells. When the nutrition conditions are correct, the yeast start to proliferate, the alpha haploid and the A haploid actually fuse together to generate a diploid, and that's how they generate more daughter cells. So the A haploid cells, an A cells produce an A factor and has a receptor for alpha. So it produces a factor, but it receives the factor from the other cells. So similarly, the alpha haploid cells produce the alpha factor. That's why it's called alpha cells. But then they have receptors for the A factor. So what happens is you have these A cells, you have the alpha cells. Once they get together, you have pheromone signaling. The pheromone signals through G protein coupled receptors. So you add pheromone, you tell the yeast to mate. Then the alpha, these pheromone signaling, then produce the alpha and A. These are the pheromones, essentially. And then they bind to the receptors on each other. It brings two cells really close to each other. And then the cells now know they need to start mating. So they stop proliferation. They stop cell cycle. They start to fuse and become diploid cells. That's how the yeast mating type switch goes. So the signaling cascade that's produced by these alpha or A factors are going through the G protein coupled receptors. So their receptors G protein coupled. The first step of activation is the same as the G protein receptor activation. So the alpha factor or A factor bind to their receptors. The receptor binds to the trimeric G proteins and allows the dissociation of G alpha subunit from the beta gamma. Now the G alpha, we already know what it does. It goes to activate adenylyl cyclase and do whatever. Today we care about beta gamma. Okay, the beta gamma get kicked away from alpha. Beta gamma actually is doing something too. It activates a MAP kinase cascade to mediate mating type switching. So the gamma subunit, again, is tethered to the plasma membrane. So beta gamma together can recruit a protein called sterol5. Ster5 meaning sterol5, because if you mutate this protein, the yeast is sterile. It cannot mate. So this protein, ster5, is a large protein. It functions like a scaffold to bring three MAP kinases cascade together. Ster11 is the MAP kinase, the RAF equivalent. It activates, phosphorylated activates ster7, which is a dual-specific kinase, like a MEK, okay? And then they activate, phosphorylated activate fuse3, which is the equivalent of MAPK. So this is a cascade, and that cascade, again, go to the nucleus, activate transcription factors, allow ster12 to bind to the promoters, and that start the mating type gene expression. Now, I don't need you to remember any of these names, okay? You don't need to remember ster5, ster11, ster7. I actually, if I don't watch this slide, I can never tell. But I need you to understand this is MAP kinase. This is MAP kinase. This is MAP kinase. And this pathway is being used it's conserved evolutionary from yeast to Drosophila to C. elegans to human, and exists in every cell type. They can mediate different downstream responses. They don't all lead to cell cycle proliferation. They can mediate many different activities, but the mode of signaling is conserved. It's all going through the 3 sets of things. So in human, there are many MAP kinase pathways, not just one. There are actually many. For example, you've heard of JNK kinase, Jun N-terminal kinase, or JNK kinase, and it's actually doing something very important, not a junk. There's P38 kinase cascade. These are all cascade, 3 kinase cascade. They can mediate stress signals when you're very stressed, when you're exposed to oxygen stress or UV. All of these stress signals will actually activate these different MAP kinase cascade so that they mediate different physiological responses. In yeast, that's even more amazing. In such a one little thing, they have many different MAP kinases. There are MAP kinases that, as we talk about, that mediate mating functions. There are ones that mediate osmolarity responses. There are ones that controls the movement of the yeast, controls differentiation of yeast. So there are at least three different type of MAP kinase cascade in the yeast. So how does the cell know which one to take, which MAP kinase cascade to take, and how does mammalian cell know which MAP kinase cascade to use when you have a certain response? The response in yeast is the scaffold protein. Okay, so different physiological responses use a different scaffold protein. The mating type use sterol5, but other osmolarity response use a different scaffold protein. And so these scaffold proteins take the right one with them so that you group the 3 right MAP kinase cascade together, so each phosphorylate each other. And the other response will put 3 other members. Certain members are shared between the different pathways. The cell do not use a whole new set of genes. The MEK and the MAPK is shared with the osmolarity pathway, but the fuse 3 is not the same. So you have a different scaffold protein that picks a different member of the 3 kinase cascade, and you can do the specificity. In mammalian cells, so far people try to look for the different scaffold proteins, and they only found one. And so obviously that's not the answer to all the problems. And so we don't know how MAP kinase specificity is really controlled in mammalian cells yet. So to show you that MAP kinase pathway is really important but can be extremely complex. And there are a lot of things that we still don't understand. The one that's activated by Ras is actually the most conserved and very traditional, the ones that we know the most. It exists in every cell type, and that particular pathway controls cell proliferation through activation of FOS. But it's not just RTKs. You can see that G protein coupled receptors can also activate this pathway. And this also gives you an example of the function of G beta gamma subunit. It's not just the alpha, it's beta gamma. And the way they did the experiment is they delete the alpha subunit in the cells. So when they add these alpha factors or A factors, the yeast, the growth and the metabolism is actually disturbed because the alpha subunit of G protein is required for all these responses, and they're gone. But as long as the beta gamma subunit is there, the mating response is normal. So we know it is the beta gamma subunit that's really important for this pathway, and alpha is doing something else. So you see that when you add a protein, when you add a ligand to a G protein coupled receptors, a lot of things can happen. You can have G alpha doing one thing, you can have G beta gamma doing another thing. And all of them come down to the central MAP kinase cascade. And again, the detailed names you don't need to remember, but you got to understand the idea of how each is activated. So you need to remember MAPK. Okay? If you don't remember MAPK, I have nothing to say. MEK, which is a dual-specific kinase, you got to know. This step always is a dual-specific kinase step. The phosphorylate activate this step. And then you have a membrane, a protein that's really close to the membrane, or it get brought to the membrane. That's the upstream cascade that activate this whole thing. And then eventually MAP kinase is one that go to activate transcription responses. So I think that's That's all for the receptor tyrosine kinase. And from the next lecture on, we're going to talk about cell growth.